Hello everyone. Today we are talking about mitosis and the cell cycle. Mitosis is when a cell divides. We begin with a parent cell and we end with two identical daughter cells. The way they are identical is in the content of their nuclei, which contains identical DNA. Cells have a life. That life begins in mitosis. Once a cell has finished dividing, it begins its new life. It then ends that life with apoptosis. Apoptosis is necessary. If a cell does not undergo apoptosis, it risks the health of the body with things like cancer. Apoptosis is triggered by a release of chemical messages from the cell that signal the body that it is time for the cell to die. Those chemical messages are called cytokines. Cyto means cell, and kine means movement, or something that causes movement. In this case, the movement that it causes is a white blood cell. This white blood cell, usually a neutrophil, releases what are called granulocytes. Granulocytes are enzymes which puncture a hole in our dying cells that make it rupture. That will cause the cell to shrivel up and be discarded by the body. So that is the beginning and end. And now we will talk about what happens in between. The cell has a cycle of life, and that begins here in G1. In G1 phase, the cell will become larger. It will also begin to create organelles. In S phase, the cell must copy its DNA. The reason it must copy its DNA is because eventually, in M phase, one cell will become two. Cells require DNA, and so if there's one set of DNA, that means only one new cell will have that DNA. To overcome that problem, the cell creates a new set so that both new cells can have DNA. And so old DNA is duplicated into new DNA. This DNA is stringy and relaxed, and so we call it chromatin. Chromatin can be read by the cell and used as information to create things like organelles. Next we have G2. G2 phase is a continuation of G1. In G2 phase, more organelles are produced because one cell must become two. In addition to having more DNA, the cell must also have organelles in order for it to work. So basically, through all of interphase, we have been preparing for the next phase by doubling everything. we have also been carrying out normal cell functions, like for example a heart beating or an adipocyte storing fat. Now we begin mitosis with prophase. In prophase, the nuclear envelope, which we've seen here, begins to break down. We will represent that with a dotted line rather than a solid line here. In addition, chromatin begins to become more condensed. If we compare DNA, if we look here in this nucleus, we see that the DNA is relaxed in a chromatin-like structure. But if we look here, the DNA takes a different shape. Chromatin becomes chromosomes. The reason that chromosomes have this shape is because of S phase. Back in S phase, one set of DNA became two. These two sets are identical, and we call each a chromatid. We also see a separation of centromeres as they move towards opposite ends of the cell in preparation to divide the cell. And finally, we see spindle fibers start to form, reaching toward the middle of the cell. In prometaphase, we see a continuation of prophase. I will not ask you about prometaphase. Instead, I will refer to it as part of prophase. In prometaphase, the nucleus continues to dissolve. The chromosomes continue to form. And these centromeres have reached opposite ends of the cell. Microtubules will start to reach out toward the chromosomes, where they will grab them in a particular place. The center of a chromosome is called a centromere. The two places that will be grabbed are called kinetochores. Kinetochores will be grabbed on both chromatids by the centromere. In metaphase, the nuclear envelope is completely gone, and the chromosomes have lined up at the equator of the cell. In anaphase, we now pull those identical chromatids in half. Because they are identical, the new cells that are being formed on this side and this side will be genetically identical. In telophase, we undo prophase, which means we do the opposite of prophase. We will reform the nucleus, and we will allow chromosomes to become relaxed chromatin. Chromosomes become chromatin. The cell also elongates like an egg. In cytokinesis, the cytoplasm is cut in half, and 2 new daughter cells are made. Cytokinesis is different for animals and plants. In animals, an actin ring, which is a fiber, goes around and around and around and around the center. It is then squeezed tight to help the cell divide. A cleavage furrow begins to form, which should be easy for you to remember. In plant cells, something else happens. A cell plate begins to form. This cell plate is an extension of the cell wall, which is a dense wall of sugar-like material that coats every cell, giving it structural stability. That cell wall will begin to grow in the center of the 2 cells, eventually creating 2 separated cells. To help you remember the phases, you can remember the acronym. In prophase, the nucleus dissolves and the chromosomes appear. In metaphase, those chromosomes align. In anaphase, those chromosomes divide, and in telophase, those chromosomes relax into chromatin. This acronym sounds like PMAT. Outside of PMAT, we have interphase, which is the longest phase where a cell carries out its normal functions. And then we have cytokinesis, which is a very short phase. Altogether, this makes the cell cycle, whereas PMAT is only part of the M phase of the cell cycle. Okay, now let's talk about what happens after division. Sometimes we do differentiation. Differentiation is the process by where one cell can become many other different types of cells. We call these cells, which have the capability to become other cells after mitosis, stem cells. There are different levels to stem cells. Some can become more than others. And so, for example, a totipotent cell would be like your zygote. This is what happens when an egg meets sperm. A few cells will result from that after 1 round of mitosis, or 2, sorry. These cells here are totipotent. This means they can become an entire person. Pluripotent cells can be harvested from the embryo. When an embryo starts to form, when a human starts to form, the zygote will divide into a blastocyst. You can take cells from here, and these cells can become any cell that you want them to in the human body. Pluripotent cells can become any cell. Multipotent cells are cells that can differentiate and can form a number of cells, but they are limited. They are usually limited to a certain type of organ. And so if we wanted to repair a heart cell, we would want heart stem cells if we were to use multipotent. The reason for this is because cells talk to each other to tell each other what to become. So the most valuable would be totipotent because they can become an entire person. Pluripotent can become any cell within a person, and multipotent is limited to the type of organ system they are harvested from. The way that a stem cell knows how to differentiate is by chemical messages. And so if this is a fat cell, neighboring stem cells know to become fat cells because of the little chemical messages that you see here. Once again, these chemical messages, which before caused apoptosis when we talked about them, are called cytokines. All right, that's it.